All right, we're doing another set of video lecture notes. So feel free to stop and pause the video or rewind as needed to get all these notes down. Of course, you can use your notes during quiz times, okay? So today we're going to be learning about DNA replication, also known as DNA synthesis. It's two different ways of saying the same thing, and you'll hear me refer to it as both, okay? And so the idea here is DNA replication is just DNA making a copy of itself. Now, if you think back to what we've learned before, when we learned about the cell cycle, remember there was G1, S, G2, and M? Well, S stood for synthesis, DNA synthesis. Okay, so this is happening during this S phase. So the DNA is going to make a copy of itself. Remember in meiosis when like the chromosomes were duplicated? Yeah, this is that. So it's some DNA making an exact copy of itself, okay? Perfectly identical if everything goes right. And just to give you a frame of reference, this takes your cells about 4 hours to do. Pretty impressive, honestly. So here, the actual process of replication is performed by enzymes, okay? And an enzyme is just a science word for a protein that helps with reactions and lots of processes, okay, in your body. Enzymes do tons and tons of things. And one way that you can identify that something is an enzyme is if the word ends in ASE, ACE. So usually enzymes end in ACE. So we're going to be learning about two enzymes for DNA replication. One is DNA helicase and one is DNA polymerase, okay? So if you remember from building your model, DNA is a double helix. It's a twisty ladder. Well, in order to make a copy of DNA, we need to kind of pull that ladder apart. We need to separate the left side of the ladder from the right side of the ladder. And that's where DNA helicase comes in. Okay, so DNA helicase is going to come in and it's going to break the hydrogen bonds that hold A and T together and the hydrogen bonds that hold C and G together. Okay, so it's going to bust open those hydrogen bonds and separate your two strands. You can kind of think of it like think of a coat that gets unzipped and think about the two halves of your zipper. Okay, and you know how like there's the little part on the left side, the part on the right side, then there's the actual zipper that does the unzipping. Okay, your DNA helicase is the zipper that does the unzipping. It is busting open your coat into like, two different flaps. Okay, that's helicase. Shown right here, it's going to be, in this case, moving to the left, and it's going to be separating all these base pairs. Okay, you can't see the hydrogen bonds here, but it would be separating them. Okay, so it's going to separate the hydrogen bonds so that the two strands of DNA get pulled open and kind of pulled apart from each other. Okay, that's your first enzyme. Your second enzyme is DNA polymerase. Okay, and a polymer just means like a chain of molecules. And so DNA polymerase is going to be making a chain of DNA. And so what it's going to do is DNA polymerase, let's say it's circled right here in yellow, is going to like hop on right here and it's going to go, oh, okay, guys, we got a C. What goes with G? Oh, we need to bring a G in. And there's going to be a whole bunch of nucleotides floating around, okay? Whole bunch of them. And so it's going to grab a G and plunk it right here. And then it's going to go, okay, next up, guys. Oh, we got an A. Okay, bring in the T. And so next the T will come in and the G and the T get attached together and so on and so on. It'll go, oh, next up we need a C. Boom, next up we need an A. And so this DNA polymerase is working on this strand to build a brand new matching strand. Another DNA polymerase is down here, okay, and it's building a new strand off of this one. It goes, okay, give me a T, give me a G, give me an A, give me a C, and it's going to line them all up and connect them, okay? So it's going to bring in those matching complementing base pairs. So we have multiple polymerases working to get this done, okay? And once your polymerases finish their job, you are going to have two complementary strands, meaning A's goes with T's, C's goes with G's, okay? And so here we are. They got busted open. You can't see it, but DNA polymerase is adding our new bases here in red. And at the end, we have two exact copies. Look at them. A, T, A, T, T, A, T, A, C, G, C, G, right? They're exactly identical. And you'll notice that in this diagram, the original DNA was shaded in blue and the new DNA is in red. And if you look at our two finished DNA strands, notice they are each half blue, half red. So when our DNA molecules are done, they are each half the original like template DNA and half newly made DNA. Okay. Here's just a nice picture of summarizing it. right? Here's our DNA in its kind of ladder form. Helicase would come in and break open these hydrogen bonds. DNA polymerase is going to come in and start matching up the nucleotides and is going to start gluing them together, okay? And at the end, we have 2 strands of DNA. They both look exactly the same.
To make perfect copies of our DNA, an enzyme first unwinds the 2 strands of the molecule by separating its base pairs. Another enzyme then attaches free nucleotides to the exposed sites. The existing strand determines the composition of the new strand. According to the base pairing rules, adenine bonds with thymine and guanine with cytosine. In long strands of DNA, replication occurs in many regions simultaneously, forming the sister chromatids required for cell division.
All right, so that was just a little animation showing you kind of what it looks like. Again, right, a cell is going to be copying its DNA so that it's preparing to undergo mitosis so that each daughter cell has the right amount of DNA, right? Just keeping the big picture in mind. Okay, and speaking of big picture, where is this happening in your cell? Where is DNA in cells? In a eukaryotic cell, it's in the nucleus. That's right. So in eukaryotic cells, this is happening in the nucleus. Okay. And obviously in a prokaryotic cell, there's no nucleus. So this is happening in the cytoplasm. As you can probably imagine, this process is more complicated than what we just made it look like. There's lots more enzymes involved. But as far as what you need to know as a freshman, we're just going to keep it simple here. If you take genetics or AP Bio or biology sometime in college, you're going to learn about all the rest of the enzymes and kind of some more in depth as far as how this all actually happens. Okay, so this has been simplified for you, but this is what you need to know for this class right now. And one thing you should know, in addition to just how the process works, is that errors happen. There's lots of moving parts here, and it is very easy to get a wrong base in. So like here's a DNA polymerase, and look, oopsies, it's put an A with a C. That's not right. Okay, well, thankfully, there's proofreading enzymes that can come in and they're checking to see are all the A's with T's and all the C's with G's. And if they aren't, like right here, the enzyme, there's different enzymes here, and one will cut off that incorrect A, and then we'll get the correct one attached. So now down here we have a G. So hooray, the error was caught. Thanks, proofreading enzyme. Now our DNA is correct. If errors get through, and this happens not very often, but it does happen that an error gets through, that you have an incorrect, an AC pairing, for example, that would be called a mutation. Okay, it's a form of a mutation. And later on in this unit, you're going to learn what are some consequences of mutations. Is it good? Is it bad? Is it just, it is what it is? We'll learn more about that later, okay? So hopefully after today, you've got a pretty good overall understanding of the general process of how DNA replication works, the 2 enzymes that are involved, and what our end products are.